In this video, we're going to finish up our discussion of the 6 types of receptors. So we've done these 3 already, and we're going to move on next to gated ion channels. We've already talked a little bit about these. Remember that gated ion channels allow passive movement. So they open up, and then the ions are just going to move in whichever direction is a negative delta G. And remember that the negative delta G takes into account the concentration gradient term and the membrane potential term. So ions all being positive or negatively charged, this term is always going to matter for gated ion channels. And remember that these can either be both positive, in which case it's not going to move in that direction, both negative, in which case it definitely does. And if one's positive and one is negative, you have to obviously actually do the math to determine which direction it's going to move in. So remember that there is a membrane potential, generally somewhere in the negative 50 to 70 millivolts range. Remember that the negative means negative is on the inside, but if we're talking about moving towards the outside, we would change the sign of that. So ions in most cells are going to have relatively similar concentrations on the in or the outside, or they're not similar on the in and the outside, but whether or not they're high or low on the inside or the outside is going to be, we're going to treat that as relatively consistent. Of course, there are different types of cells, but generally speaking, potassium is high on the outside and low on the inside, and sodium, calcium, and chloride are all higher on the outside than they are on the inside. We've talked about how the sodium-potassium ATPase is one of the things that is keeping the potassium high and the sodium high. The potassium is high inside and the sodium high outside. There are similar things that are going to affect the calcium and chloride concentrations. I just want to clarify that the fact that the sodium-potassium ATPase is moving these things in, and then an ion channel might open that causes them to move in the other direction, does not make the ion channels secondary active transporters. So to be a secondary active transporter, you have to use this, but then really be a co-transporter of some sort, that you're bringing something else in against a gradient that is not related. So because they're just ion channels, it's not considered secondary active transport. So another thing about ion channels is they're usually only open for a very short period of time, but they're considered to not be saturatable, which means they do not have sort of the Michaelis-Menten type, there's a Vmax sort of thing. They can go through as fast as, there's no limit to as fast as they can go. Realistically speaking, of course there is a limit, just based on how fast the sodium ions can just get here and move through, but practically speaking, we do consider there to not be an official limit, because they're not a limit for the same reasons that there are for enzymes or active transporter, or not active transporters, but anything with 2 gates, so transporters versus ion channels. So again, remember, passive movement. So the main place we're going to talk about gated ion channels is signaling in neurons. We will see more of these in the future, especially in term 2. We'll see a few of these, but the neurons is probably the most famous use of them. So just a little bit of background here about how the neuron works. All right, so we have this is one neuron cell, and neuron cells are sort of long, and then they're going to meet up with another one right here. This is called a synaptic cleft. This is a space between 2 cells. And what's going to happen is this neuron needs to pass a signal on to the next neuron. There's also going to be the signal passing down the nerve cell, and that's also going to be using ion channels. All right, so starting up here, there are a whole bunch of ion channels along the length of the neuron. And so there are sodium channels all along it. Remember, sodium will go in when you open them up. And these are voltage gated, so that's going to mean that they open in response to a change in the membrane potential. That can be a hyperpolarization where it makes it more negative, or a depolarization where it makes it less negative, or sometimes even reverses and it's positive inside. But it's in response to a membrane potential change. And there's also potassium channels along the neuron. This is showing them being on two different sides. Just to be clear, that's just to kind of keep you oriented. It's not like there's one side of the neuron with this and one side of the neuron with these. Potassium channels, of course, are going to move out of this. Like the potassium is going to move out of the cell, right? So, and at the very end, only at the end, do we have some calcium channels that are also, all these are voltage gated, and the calcium channels are going to allow calcium to move in. So the reason these are at the end, but these are all along, is we are going to send a wave down the neuron where one sodium channel is going to open up in response to depolarization. So depolarization, remember, is putting more positive things on the inside or more negative things on the outside. So this sodium channel opens up in response to the membrane being depolarized. But as sodium moves in, it further depolarizes it. So that is what's going to then pass the signal down here, because now this one senses that there's a depolarization in the membrane, and then it opens and passes things through, and then all the way down. Because this depolarization of the membrane is relatively small, it's in a small area. It doesn't depolarize the whole cell. It depolarizes sort of that chunk. So what's going to happen is at the end, we're not seeing the end up here, but something starts the signal, and then these sodium channels start opening and passing the signal down the neuron. However, we obviously can't leave those open forever, or you wouldn't be able to have any new nerve signals. So the purpose of the potassium channels is to repolarize the membrane. So they open also in response to the depolarization, but as they open up and the potassium flows out, that is going to repolarize the membrane. The calcium channels are of a different purpose, so they also open up in response to depolarization. So when this thing that's passing through the sodiums hits the calcium channels, it will open that up there. And what happens now is calcium comes in, and it is going to cause the release of some vesicles that are in the nerve cell that contain acetylcholine. This is a small molecule. So it causes them to be exocytosed. They sort of move over here, and it pushes out this calcium in there, and the acetylcholine in there. So let's actually look over here, because this kind of looks like that's what's coming out, but it's these little red molecules, the acetylcholine, that's coming out. Acetylcholine is a neurotransmitter that moves across the synaptic cleft and binds to an acetylcholine receptor, which is also an ion channel. This is a ligand-gated ion channel instead of a voltage-gated one. When these open up, that lets sodium, potassium, and also sometimes sodium, calcium, and sometimes potassium in, but it lets positive things in, right? Potassium not likely to come in, right, because it would be going out. So it's mostly sodium and calcium, but sometimes potassium can go through. That's going to let these sodium and calcium in, and that's going to start the action potential of that wave of depolarization, because this first step of sodium and calcium coming in is going to start depolarizing the membrane, and then these sodium channels keep it going, and then eventually we get up to here. So there's really two waves. There's the sodium channel wave, and then just following it is going to be the potassium one. All right, so it's a lot of different things. That's 1, 2, 3 different types of voltage-gated channels, and one type of ligand-gated channels. So we're going to just refresh a little bit on how the voltage-gated channels work. We saw a voltage-gated potassium channel earlier, and here we're just going to see the structure of a sodium channel. It's pretty similar, although sodium being smaller, it's going to not let potassium in just because it's just a little bit smaller. So it's got a bunch of helices around here. These little pink loops right here are selectivity pores. That is what's going to be just the right structure, just the right size to only let sodium through. And so then it's also got these voltage sensors. These have a bunch of things like arginines that can pick up positive charges and change their protonation state and then change the way they're configured if there's a difference in membrane potential. And then that just opens up the pore and it can go through. There's also an inactivation gate. And what this is shortly after the channel opens, even if the membrane potential is still depolarized, the inactivation gate moves in front of it. So it basically turns it off. This is a type of desensitization where it turns it off so that it won't keep going. This ensures that the action potential is unidirectional, because what we don't want is maybe halfway down the axon, some sodium channel opens up, depolarizes the membrane, and then the channel just ahead of it reopens up again, right? Then you'd have all these action potentials going up and down. That wouldn't make sense. So these inactivation gates make sure that it only moves in one direction. So once you've been opened, shortly after that, it's going to close back up, and then it takes some time for this inactivation gate to sort of move back out of the way so it can be reactivated. This is just a better picture of that activation gate and the voltage sensor. So when the voltage sensor changes its protonation state, it changes configuration, and that causes the activation gate to move, open up so that the sodium can flow through. So those are the voltage gated channels. That's true. That's the sodium, the potassium, and the calcium channels. The nicotinic acetylcholine receptor is the first ion channel in the pathway, and this is a ligand-gated one. So it does not rely on the membrane potential. What it relies on is acetylcholine bound there. All right, so this has these M2 helices, and there's 5 subunits, each that has 4 alpha helices that go through, and each one has an M2 helix that sort of points into the pore. And this is going to be turned on, opened up, when 2 molecules of acetylcholine. So we've actually seen choline before, right? Choline looked sort of like ethanol with this amine and then 3 methyl groups on it. This is acetylcholine, so we put an acetyl group on the OH. So it's just a small molecule. And when this binds, so 2 molecules are going to bind into this channel. When that happens, it causes everything kind of rotates, and that's going to open up the pore. So this conformation shift on binding, this is what happens. So in the closed configuration, those M2 helices are what's sticking into the pore are these kind of bulky hydrophobic leucines. And so there's not really going to be much in there. Even the water's not going to want to go in there because it's leucines, right? Leucines are sticking pretty far out into the channel. That's going to close the channel. This almost just looks like a regular folded protein. When the acetylcholine binds, that causes everything to rotate a little bit, and that causes these little smaller polar residues, something like maybe a serine, to be pointing into the channel. So they're A, smaller, so there's more space, and B, polar, so that water and the ions can flow through. So one frequently confusing thing that people have is they think that the acetylcholine receptor moves a cell, like acetylcholine flows through it. All right, so it's not, right? That's why it's called the acetylcholine receptor. The acetylcholine binds, and then sodium and calcium flow through, starting the action potential. All right, so that's gated ion channels. We're going to briefly talk about these adhesion receptors. So adhesion receptors, there are a number of them, but the most famous ones are called integrins. And so the way adhesion receptors work is they have a subunit on the outside of a cell and a subunit on the inside of the cell. And on the inside of the cell, it's attached to the cytoskeleton. So in human cells, it's mostly going to be actin, polymers, or microtubules, so proteins that are helping hold together the structure of the cell. And on the outside, they can bind to various things in the extracellular matrix. And so integrins are active when they are dimers, and they come in a whole bunch of different variants. I think there's something like 18 of one type and 10 of another or something like that. And they can come together in different pairs, and that just affects what sort of things they're going to bind to. What's interesting about the adhesion receptors is they can signal in both directions. So all the other receptors we've seen, a signal comes in from the outside of the cell and then causes a change on the inside of the cell, right? And that can happen for integrins. So there might be a change in the collagen in the extracellular matrix, which causes the integrins to bind to it, and then a little bit of a change in here. And since it's bound to the actin, that causes maybe the actin to change. But it can happen the other way around too. So a change of something in the cytoskeleton, or binding to this protein called talin, it can cause the other direction happen. A change in here can cause a conformation change out here and cause the integrins to either bind or not to bind to something in the extracellular matrix, which can cause just a difference in what it's bound to. Adhesion receptors, I think, they're a little bit different because they don't always lead to these long pathways and then a really downstream effect. They're a little bit more direct, so with one we're not going to spend too much time on them. But they're really important in cell-cell interactions and cells interacting with the extracellular matrix, which I think we tend to sort of forget about. It's easy to ignore it and focus on what's going on inside an individual cell. But how the cells are interacting with other cells is also really, really important. All right, so the sixth and final class is down here. You may have missed it because it wasn't up here on the surface of the cell. And the reason for that is that these are receptors that are actually already in the nucleus. So these have to have ligands that are able to pass through the cell membrane, the plasma membrane, and move into the nucleus and then bind to their receptor. So we've seen some other pathways that eventually ended up in stuff happening in the nucleus, but the initial receptor was still out on the plasma membrane. In this case, right, like here with the kinase cascade, right, in that. In this case, what's happening is even that initial signal passes all the way through instead of making second messengers or kinase cascades or something, and goes all the way to the nucleus and then causes the transcription of mRNA, eventually leading to proteins. So the most common class of molecules that are going to be nuclear receptors are going to bind steroids. So things like really the steroid hormones, testosterone, estradiol, cortisol, things like that. And the reason for that is that they're very hydrophobic. So they are able to pass right on through the membrane, and then they can be transported into the nucleus. The nuclear membrane is a little bit more porous. That's why they're showing it like that, where they can just bind directly to nuclear receptors and cause gene transcription, right? So this also doesn't really have much of a amplification cascade. Now, you can argue that even just the action of transcription and translation is a little bit of an amplification, but it doesn't have anything before that. So these often are not going to be things that have to act very fast. So if you think about it, the action of, say, testosterone, which might cause, say, male puberty, or the action of progesterone or estradiol that might affect women's menstrual cycles, that's not, we're talking days maybe for that to happen, or months or whatever, right? It's not the sort of thing that you have some sort of signal and you need your estrogen to spike in like a half a second. So it's not as important to go through that that amplification cascade. It's not the same as having, to have an epinephrine spike or something like that. Just the speed is not as important. One thing I actually do want to clarify, steroids can absolutely bind any of the other classes as well. So steroids can pass through, but there are definitely examples of steroids that can act through GPCRs or RTKs or things like that. And just because they can doesn't always mean that they do. So if we had any steroids that were going to act in faster ways, those probably would not be acting through this. They would probably be acting through a more normal plasma membrane receptor. So here's just reminding you what those steroids look like. So this is estrogen and progesterone. These are two hormones that are particularly important for females. And just a reminder of the fact that drugs can often be designed by making them look very much like the native ligand or signal, and that would be an antagonist, right? Because it would bind, well, I guess in this case they're antagonists. They would bind and they stop the native thing from binding and starting the pathway, because they're going to bind there but not start the pathway. And this is just two examples. So tamoxifen is a drug that is often used in breast cancers and ovarian cancers and things like that, things that are estrogen sensitive. So there are a number of cancers where estrogen is actually causing a problem. And so cancers like that, you can use this tamoxifen, which binds to the two estrogen receptors and is an antagonist. This mifepristone, which is also called RU486, you may have heard of. This is a abortion drug. It's not the morning after pill. It's actually an abortion drug for early pregnancy. And the way this works is it is a progesterone antagonist. Progesterone is a hormone that is really important in pregnancy. And if you're not getting enough progesterone signals, pregnancy cannot continue. And so what this does is it binds to the progesterone receptor and essentially blocks, even though there's progesterone there, it's an antagonist, so it's going to block the receptor. You can also see that these are quite nonpolar as well. For any antagonist or agonist to bind to a nuclear receptor, it also has to be able to pass through the membrane, get into the nucleus, and so something like tamoxifen wouldn't work if it got stopped by the plasma membrane. All right, so that is the end of our 6 classes, and then just the end here of the chapter, just going to go through some review on why really this is all about phosphorylation. We saw a lot of phosphorylation and signaling. We saw a bunch of kinases. Phosphorylation is often used to turn things on and off. So just that's very important. So we're just going to clarify a little bit of this stuff. It's one of the reasons is it's quick and it's reversible. So especially for pathways that may turn on and off and on and off and on and off regularly often. So something like insulin, every time you eat something, your body adjusts that pathway. So we don't want to be bothering to destroy proteins or whatever, so we just phosphorylate them to turn them on and off. So we've seen a couple examples already of these proteins or domains that are going to bind to phosphorylated residues. The one that I really focused on earlier was the SH2 domain. But there are also PTB domains, which just stands for phosphotyrosine binding domains. And so these are these are going to be kinases. They're going to put this is particularly talking about phosphotyrosines versus phosphoserines or phosphothreonines. So these phosphotyrosine kinases are going to put the phosphates on. Phosphotyrosine phosphatases are going to take them off. So these are so important that there are just hundreds of them. So if you look, this is humans. There's over 100 known proteins that have SH2 domains in them. And there's about 90 different known proteins that are phosphotyrosine kinases. So that's a lot of different things. And you can see this is pretty common. Lots of different organisms have a lot of these things. So the reason for that is just because it's a system that works relatively well. And so evolution often doesn't reinvent the wheel. It'll just use things over and over. Just to be clear, something like a PTB or an SH2 domain, this is not it's not identical. In fact, different SH2 domains do not have really any sequence similarity to each other. It's just that they have the same function. One useful thing about these is they can be easily auto-inhibited. We already saw this one time, but the reason for that is their substrates are proteins themselves. And so if you have here is the active form, where here's the substrate that it's going to be acting on. This is so Sarc is a kinase. So Sarc is going to be putting phosphates onto this tyrosines. And Sarc also includes an SH2 and an SH3 domain. SH3 domains are just another type. They bind to different things. What we can see here is that in the time when it's going to be active, this SH2 domain is binding to a phosphotyrosine that's already in the substrate. But also on this enzyme, there is a tyrosine over here that can get phosphorylated itself. And if this is phosphorylated, what happens is the whole thing loops back around. The SH2 domain binds to the phosphotyrosine in its own protein, and that just changes the configuration. You can see it kind of like pulls this direction. Changes the configuration so we no longer have an active site. So we can't act on this substrate, even if it's around. Over here is just another example of how it doesn't have to have an SH2 domain to have auto-inhibition. GSK3 is the enzyme that's going to phosphorylate glycogen synthase, and it has an active site, and it also has another site for specificity where a phosphoserine on the glycogen synthase binds to sort of put these guys in the right spot. So it has a portion of it. This is not part of an SH2 domain, but it's just a portion of the protein that binds to a phosphoserine. And we can see that there's also a serine up here in GSK3 that can be phosphorylated, and that is going to loop back around and inactivate it. So we talked about how phosphorylation can either activate or inactivate things. With kinases like this, often it is this is the reason that being phosphorylated will inactivate it, because you can have some sort of looping back around. So remember where I said there's modularity? There are even proteins that have whole bunches of these different modules. They might have some SH3 domains and also these SH2 domains that have different binding purposes than these. There's other types of domains. So I don't want you to stress too much about the details of this, but just the fact that we can kind of put these modules together. These are domains, which remember means that they can fold independently. And so it makes it a little bit easier to potentially sort of stick them together in a protein. Phosphorylation can also affect these scaffold proteins. Remember, scaffold proteins can be useful to hold everything together. So this is the RAF-MEK-ERK pathway that we saw in one of the RTKs. This is the MAPK cascade pathway. And there's also a scaffold protein that can hold these three together so that they can very quickly activate each other. However, if you want to inactivate this pathway entirely, you can phosphorylate the scaffold protein itself, which causes RAF not to be able to bind. And it can still come and phosphorylate MEK, but it's just going to be a little bit harder for it because it's not being held in position and might float around all sorts of other places. So phosphorylated residues, really good at modulating binding interactions, basically. So that's the end of this lecture. We are skipping some portions of Chapter 12, so if you're reading Chapter 12 and there's something we didn't talk about at all, that's because we're skipping that, and more than normal, that is. And the last lecture on Chapter 12 is just going to be giving a few more examples of interesting pathways. So it's not really going to be really new information, it's just going to be new examples of things we've already seen.